Welcome to the 1 Minute Apologist. We interview the world's leading apologists to provide credible answers to curious questions.
Jonathan, give our listeners an example of irreducible complexity.
So irreducible complexity is where you have a system made up of many or several well-matched interacting components, the removal of one of which renders the system non-functional. And so the issue is, how would you build a system of that sort up in terms of a Darwinian incremental process? And if you look at cell biology, biology is absolutely full of such systems. One example, there's many that could be given, is in bacteria. Bacterial cells divide by a process known as binary fission. And basically, in a broad-shaped bacteria, the bacterial cell will elongate to about twice its original length. There'll be there's a Fitz-Z ring that polymerizes around the division septum and it bifurcates into two daughter cells. Now, the Fitz-Z ring, which assembles along the division septum in the middle of the cell, it has to be tethered to the inner membrane of the cell. If the tether is missing, then the cell can't divide. The Fitz-Z ring has to be tethered. So how do you get the evolution of this Fitz-Z ring with no tether? And moreover, when the bacterial cell elongates, it has to literally sever the peptidoglycan cell wall, which is done by enzymes known as autolyses, which have to be very tightly regulated and controlled. And the cell wall has to be resynthesized as the bacterial cell grows, because what happens if it's not resynthesized is that because of the osmotic pressure, the cell bursts or lyses, if you will. And so it's put back together by a process known as transpeptidation or peptidoglycan cross-linking. There's a protein called lactoprenol, which shuttles the peptidoglycan precursors across the cell wall. There's penicillin binding proteins, which are involved in the peptidoglycan cross-linking process. And what's interesting is that these penicillin binding proteins are themselves a target for penicillin antibiotic, because what happens is if the transpeptidation of the peptidoglycan cross-linking fails, and the cell bursts, then the cell dies, and so that's why it's a target. But how would you put together through a Darwinian process, a mechanism to, in a very coordinated fashion, sever, rebuild the peptidoglycan cell wall? Because if you simply develop the process for breaking the peptidoglycan cell wall, then you're dead until you have the mechanism for rebuilding the peptidoglycan cell wall that has to be done in a very coordinated fashion.